And now it's time to invite Anna to join us and see how it is that all of those validations, maybe not perfect, whatever LLM, how is it that we can use them to adjust our study result? How it is that this is done currently?
Okay. Hi, everyone. So some of you were expecting a break, but instead you're going to get quantitative bias analysis. So here we are. I apologize in advance. I can see some of my fonts are a little bit wrong. But yes, so we've heard a lot about how we can measure the validity of different variables in databases. So assuming we've done that, we have our validity indices. What do we do? Well, so quantitative bias analysis is one option. Before I start, I just want to give my thanks to Ian Douglas and Marlene Boken, who both helped with slides and the work kind of leading up to this. Okay, so I want to start off by kind of taking a little bit of a step back and think about how we handle error in epidemiological studies. So if we start with random error, so this is something that I think we all do quite a bit of. It's sort of lesson 2 in your epidemiology degree, is some kind of quantitative assessment of random error with confidence interval with p-values. I think for most of us, it would be completely impossible to imagine publishing a study without quantifying random error. Confounding, another thing that I think we are quite good at thinking about, so we have DAGs that allow us to identify different variables. We have lots of different methods that help us adjust for confounding. And then we get to misclassification. And sorry it cuts off there, but basically, traditionally, I think we've done some kind of qualitative assessment of misclassification. So we might say something, something, biased towards null. And effectively encouraging researchers to don't worry and instead be happy. So I think it's also fair to say that the status quo needs improving. Because the truth is that although we have this heuristic that non-differential misclassification biases towards null, that is true in some instances, except for when it doesn't. And I don't have time to go through all of the different scenarios where that doesn't hold true, but there's a really nice paper by Weiland and colleagues that lays that out. And we've also heard that it's important when you're considering absolute risks, but I would also say that when you're thinking about relative risks, the extent of bias towards null actually also matter. So if you imagine a relative risk of 1.5, you have some bias towards the null, you might think about it, but does that mean that your true relative risk is 1.6 or that your true relative risk is 55? You know, so from a sort of regulatory perspective, that's probably something that you want to be able to know because the decisions you take will depend on this. It's also true that we are quite bad at intuiting how large these kinds of biases are, and there's lots of different work from other disciplines in psychology sort of laying that out. So what is quantitative bias analysis? So quantitative bias analysis is a set of methods that allow us to quantify the direction, magnitude, and uncertainty of systematic error. I think it's fair to say that QBA is increasingly used, although not as often as I think we would like to see it used. We can use QBA to correct for unmeasured confounding, for selection bias and misclassification. And this graph just shows you the breakdown of the different scenarios in which it's used. So actually, most frequently for misclassification. So when we think about QBA for misclassification, effectively what we're trying to do is we're trying to adjust an estimate of effect for misclassification. We do this by setting some assumptions called bias parameters. And for misclassification, the bias parameters we can use are the sensitivity and specificity. That's typically what we would tend to use in QBA. And there are also certain scenarios that you can use the MPV and PPV in. Where do we get the information on these? So, I mean, ideally, we've heard a lot about how to conduct a fantastic internal validation study. So hopefully that is what you have. If not, then I think there's sometimes external validation studies. If you're happy to assume that that applies to your population, you could use that data. Or you could use an educated guess. Oh, you can't really see, but these numbers effectively just show that the educated guess is in about a quarter of different cases. There's also different methods of QBA, so it's effectively a set of different methods, ranging from simple, where we effectively just use a single value for our bias parameters, to probabilistic, where we set a distribution for those. So We've seen a lot of 2 by 2 tables today, so I'm going to go through this very quickly, but I wanted to kind of give you an intuition for what QBA is and how it works. So I think a lot of us are quite comfortable with the idea that if we know the truth, we can use the sensitivity and specificity to kind of get at what we would observe in a database. So if we think about the exposed cases here, I think most of us would intuit that we have some false negatives, and for what we observe in the data, we need to remove the false negatives. We also know that we have some false positives, so to get at what we observe in the data, we kind of want to add them in. You can put those together, you end up with a little formula for what you observe in the data, and then, because we obviously don't know the truth, we can actually just rearrange that equation, and we end up with effectively a formula for how we can get at the expected truth. So you do this for all of the different cell counts, you end up with what is effectively your bias formulas. So the reason I'm showing this is not because I think we're all ready for more maths, although maybe some of us are, but just because I want to give you kind of an intuition for how these methods work, because I think sometimes there's an assumption that they're really complex, but in its essence, they are really intuitive and just using your intuition about sensitivity and specificity to do these kinds of adjustments. So we also have something called probabilistic bias analysis. So in PBA, we use Monte Carlo sampling methods to effectively generate a distribution of bias-adjusted effect estimates. The way we do this is that we assign probability distributions to bias parameters, and then we sample from those. We then take the sort of individual value we sample and correct our 2 by 2 table, and then we do this very many times. And you can do probabilistic bias analysis either on summary level data, so effectively the 2 by 2 table that I just showed you, which is quicker but has the downside of not allowing for confounder adjustment, or you can do it on the individual patient record level. So effectively what that does is simulate different outcomes for each record in your dataset, and that has the benefit that you have this analytical flexibility so you can adjust for confounding. Quickly about incorporating uncertainty. So I think the point about quantitative bias analysis is that it's a means for us to quantify both random and systematic errors, or at least get us to think both about random and systematic error. In simple bias analysis, I've showed you all the formulas. You might be tempted to just kind of go and apply them and then think, oh, I have a 2 by 2 table. Let me calculate my confidence interval. So that you can't do that because that doesn't take into account that kind of added uncertainty. So there are separate formulas, but what I think most often happens is that in a simple bias analysis, people just present point estimates. So you really only check how much your point estimate varies. In probabilistic bias analysis, on the other hand, we simulate random error around the bias-adjusted estimates within each of our simulations. So we're actually able to get an estimate of uncertainty or total error that incorporates both the systematic and the random error. I wanted to give a quick example of how we used these methods. So I want you all to kind of throw your minds back to COVID, first COVID lockdown. There was lots of interest during this time in how different drugs affected the risk of COVID. One of the studies that I was asked to do was to look at inhaled corticosteroids as a potential kind of treatment or repurposing candidate for COVID. There was, I'm sure lots of you worked in this space as well. There was a lot of time pressure, so we're sort of having to do these studies in weeks rather than months. We ran the study and we actually found a harmful effect. So inhaled corticosteroids kind of increased the risk of COVID, and later randomized controlled trials actually found a null or a potential protective effect. So that's not fantastic because it's effectively the opposite of what we were hoping for. But luckily, we had a PhD student join our team who has been looking into effectively the impact of different errors on these results, thinking about, well, can we use some of these methods that we've learned since to learn what we could do better next time. So one of the biases or potential biases we were interested in looking at was outcome misclassification. So this graph here shows the amount of COVID deaths over time in the UK. You can see this huge spike kind of in April in 2020. And you can also see that there's a lot of kind of excess deaths that aren't technically COVID deaths, but we think they are. So we are effectively concerned about misclassification here. So in more technical terms, what we're worried about is low sensitivity of our estimate of COVID deaths. So we think we might be missing over a fifth of COVID deaths. But we were also actually worried about reduced specificity. So in the UK at the time, we had really limited testing capacity, and we were concerned that some of the death certificates that listed COVID might not actually be due to COVID. And two things to note here is that what we were worried about was effectively misclassification of the cause of death, not whether or not someone died, and not when someone died. So just the cause. So we set out to figure out whether this effectively was an issue. So to do this, we needed some bias parameters. And what we actually didn't have a validation study. I realize this is ironic because it's a workshop about validation studies, but maybe we can discuss why we didn't have this available at the time. So what we had to do was fall into that kind of educated guess bracket. And we instead used sort of three different sources of publicly available information on COVID deaths, triangulated them, and were able to get some estimates for what we think the sensitivity and specificity was over time. This just shows the results. So the study compared uses of inhaled corticosteroids to uses of effectively an active comparator in the population of COPD patients, which is LABA/LAMA uses. And you can see, so before we correct for anything, we find a harmful effect as we had learned to come to expect. On adjustment for confounding, this moves a little bit towards the null, but not completely. Simple bias analysis, this is interesting, brings us right back to where we started, but essentially shifts the point estimate further away from the null. We find similar things for summary level and record level PBA, but you can see we now have the confidence interval, so we have increased our uncertainty. And then when we conduct record level PBA and incorporate the confounding adjustment, we move a little bit closer to the null again. That was under the assumption of non-differential misclassification. We also wanted to look at differential sensitivity. And what we were worried about here was effectively if we were missing a lot more COVID deaths in one of the treatment groups than one of the other treatment groups, could that potentially bias our results? And you might think, well, that's not very likely, but we were worried that some of these patients might be, for example, being cared for in care homes and therefore might be more or less likely to have COVID recorded on their death certificates. So this really nice heat map that Marlene has made shows a sort of range of scenarios. And what we were interested in is effectively how bad would the differential sensitivity have had to be for us to kind of miss a protective effect. And that's a little green corner. So that essentially shows that we would need almost perfect sensitivity in the inhaled corticosteroid arm and very low sensitivity in the LABA/LAMA arm, so below 60%. So how do we interpret this? Well, we missed a lot of COVID deaths, that was true. But in the context of high specificity, non-differential reduction in sensitivity did not matter. We've heard this several times today. We saw this small shift kind of away from the null that was driven by the reduced specificity. But what we did do when we did the sort of probabilistic bias analysis is that we increased our uncertainty. So I think this is actually really important because that's kind of what we want to do in these methods. We want to acknowledge that we are a little bit less certain than we think about these things. It's also quite nice to be able to quantitatively show this to people who might not have this sort of intuition for how different biases work, and say, well, you know, you're not really capturing any deaths. That's very bad. But here we can say, well, actually, didn't make a huge difference for us. We also, this is a qualitative interpretation, but the extent of differential misclassification that would be needed for us to kind of miss an effect we thought would be quite unlikely in the context of an active comparator. We have these sort of hypotheses around unmeasured confounders, but it's quite hard to imagine that the coding of death would vary that much, I think, by treatment. And if you want more detail, this work was published really recently, slight advertising for that. Okay, so I know it's late, so I have a few recommendations. Some of them we've heard before, that if you do a validation study, please do it by exposure. That's very helpful. I wasn't actually sure, Shabby, if you were planning to do a validation study or a comparative sort of treatment study. But if you were planning to do sort of comparative treatment studies, here's some recommendations for QBA. So what I think is important is that you pre-specify your QBA. So first of all, when you're going to run it. So there's certain scenarios when QBA isn't that useful. So if you have a really imprecise estimate, then just adding in this sort of systematic error doesn't really make a huge difference. And also have a think about how you will define your bias parameters. Will they come from an internal validation study, an external validation study? You might be wondering, well, you've presented 3 different methods effectively of QBA, which one should I use? I think it's quite neat to be able to incorporate this uncertainty around the bias parameters, so I would probably recommend probabilistic bias analysis. Oh, sorry, my slides have cut up, so I'll just tell you. But if you are doing a distributed analysis, for example, in a sort of VAC for EU kind of context, I think the summary level analysis is quite nice because you can do it yourself. Although if confounding is likely to be a concern, then you're stuck with kind of the record level approaches, which are a little bit more computationally intensive, not insanely so. We're not talking weeks to run, but we are talking kind of overnight. Okay, and then some questions I think we have been pondering in our team. So when we set out to kind of apply QBA to this, what we found was there's a wealth of resources, right? There's books, there's R code, there's macros, there's Excel spreadsheets, but most of them are sort of set up for quite simple scenarios. You have a binary exposure, you have a binary outcome, you have an odds ratio, and you want to correct it. So we actually ended up changing or tweaking our study design a little bit because it was easier to kind of fit it in here. And something we've been wondering is, well, how do you move to kind of more complex scenarios? So what if your bias parameters are varying with confounders? What if they vary over time, which most probably they were in our scenario, and we kind of ignore that. And we've also heard about this sort of delay issue or time to event outcomes. So we assumed just the course was misclassified, but what if we'd been looking at COVID tests? Then probably the sort of recording time of that might also be misclassified. We've also spoken a bit about kind of transportability methods, when you might be able to transport sensitivity and specificity. I think there's been some really exciting work in that space, and I think it would be really nice to see more applications of those kind of methods here. And then I guess to finish on this, just how does QBA compare to alternative methods? So if you run an internal validation study, you have your gold truth. Should you be using QBA? Could you use multiple imputation? Is this a missing data problem? So I think there's some of my questions, just a few other ones as well, but I think I'll leave it at that. And let me see. Thank you very much for your attention.
Thank you so much, Anna. So finally, we are starting to incorporate in our analysis what we know, but of course there is still a lot missing. Any burning question, please, Shabi.
Well, yes. So you're anticipating the second paper that came out of this work, which I don't want to spoil it because we're hoping to present at ISPE. But oh, sorry. Um, but yes, of course. So the aim was kind of that we'd look at all errors kind of systematically. So we're considering misclassification, we're considering selection bias, and we're considering confounding. And we are actually seeing really interesting results when we look at different methods to control for confounding. So, for example, applying high-dimensional propensity scores, which we think in this case, you look really skeptical, Rosa, but in this in this case,
All of them together with that moment
um, but yes, so that is something we've also looked at.
Thank you, Anna.